Kia ora koutou katoa. Can everybody hear me? Fantastic. Just wanted to thank you all so very much for coming to this UC Connect lecture tonight. Really excited to have a fantastic audience. Really looking forward to a fun hour or so here talking about space. Just really quickly before we get started, just a little bit of housekeeping. So you are in the C block lecture theaters here at the UC campus. Toilets, if you didn't see them on the way in, are here on the ground floor, just off to the right, well, to my right, just outside these doors here. In the instance of an earthquake, stop, drop, and hold, just stay put until shaking stops, and then exits. We have main exit here. We also have exit up at the top of the lecture theater as well. Just follow the signs around to an exit. If there's a fire or another emergency, we've got volunteers that will help lead us out to make sure that we're all safe. Other than that, I think that's most of the housekeeping that I'm supposed to be taking care of. So without further ado, thank you so very much, like I said, for coming here to tonight's lecture. So I'll be giving about a 45-minute talk, and then we'll have plenty of times for questions at the end. And if, for those of you that ask questions, I have mission patches, so just putting that out there at the beginning. Excellent. So thank you very much again. So I'm Sarah Kessans. I'm a senior lecturer above the bar here in the School of Product Design in the Faculty of Engineering. You might be able to tell from my accent, I'm not a native Kiwi. I've been here right at 12 years, so just celebrated 12-year anniversary here in New Zealand. I came to Aotearoa for a 2-year postdoc, and they haven't kicked me out yet. So thank you all for making Aotearoa my home now. It's really lovely to be here and to be part of the community, especially here in Otautahi. It's very much home now. So I've been here at the University of Canterbury for 12 years as a postdoc. Then going through my career, I'm originally a biochemist, so it might be interesting to hear a biochemist giving a talk on space, but hopefully by the end of this lecture I'll convince you of the value of biochemistry and scientific research in microgravity. So just real quick, just so I can get a little bit of an understanding of our audience here, how many of you have seen a rocket launch or have launched a rocket yourselves? So we've got a few. How many of you would like to see a rocket launch? Fantastic. All right, you're in the right place. Very good. Just a couple other questions. How many of you would like to work on rockets or work in the space industry? Anyone? Very good. For those of you online, we've got some really great audience members. All right, so last question just to kick us off. How many of you would like to sit in that rocket on the tippy top of that little rocket and go to space yourself? Excellent. All right. Fantastic. Very good. So again, you're all in the right place. We have a very friendly audience here, very much shares my passions and my excitement. So I don't think it'll take any convincing that space is really exciting, but what I really want to talk about is what we're doing in space and what the value of scientific research in space is really all about. So just to start off, Alan McDermott was one of New Zealand's Nobel Prize winners. New Zealand born. And one of my favorite quotes of his is that science is people. He would not be able to get the Nobel Prize without incredible teams of people around him. And similarly, space only happens because of people and the teams that are doing them. So I have the honor of giving this talk tonight, but really the talk that I'm giving is the amalgam of a lot of work by a lot of amazing people who I'm really, basically really honored and fortunate to call friends and teammates. So a lot of the work that I'm going to be talking about over the course of the next sort of 40 minutes or so are because of these amazing people from around Aotearoa, from around the world. So as we're going through this talk, science is people, space is people. It's not, I just get to talk about the work that these amazing people do. All right, so before we begin, this is a lecture. There will not be a test, but there might be pop quizzes throughout the lecture, so just everything that you're taking in, just keep that in mind. So what is microgravity? So when we're talking about microgravity, and what I'm going to be talking about here for the next sort of 40 minutes or so, is in the microgravity environment. So I want to show of hands, microgravity is there's zero gravity. Is it no gravity whatsoever? All right, again, we're in the right place. Very good. So microgravity is not the absence of gravity. So when we're talking about the microgravity environment, mostly what I'm going to be talking about is the low Earth orbit. So within about 400 kilometers is the orbit of the International Space Station. And the gravity at 400 kilometers above the Earth is actually about 89% of Earth's gravity. So there's it's quite a bit of gravity. So when we're talking about microgravity, it's not the absence of gravity. So essentially what's happening to the International Space Station as it's orbiting, again, this 400 kilometers from Earth, is actually free falling in the vacuum of space. So it's outside of the Earth's atmosphere, and so it's essentially free falling back to Earth. It's just traveling really fast. So it's traveling at about 27,580 kilometers an hour. So it's going pretty fast. So basically orbiting the Earth sort of every 90 minutes, getting a sunset and a sunrise sort of every 45 minutes. Just absolutely incredible. But there's very much gravity in this low Earth orbit. But what's important for us, and what we really want to think about when I'm talking about microgravity and the microgravity environment where we're doing all this amazing research, is thinking about microgravity as the condition at which objects appear to be weightless. So if you've ever seen astronauts sort of flying through the International Space Station, they look like they're weightless, but they're actually falling back to Earth. They're just traveling really fast, such that the International Space Station is staying in orbit. But this microgravity environment is really important for the research that we're able to do. So a little bit more in depth. So just again, it's not the lack of gravity, it's just where objects appear to be weightless. But there's some really cool physics going on in this microgravity environment. So I'll just go through a few of these. We're not going to get into lots of equations and maths and things, so that's not going to be on the pop quiz later. But some important aspects and sort of physical aspects of the microgravity environment have to do with, obviously, that microgravity. So here on Earth, we obviously have Newton's laws, we have gravity. If you have an apple on an apple tree, it's going to fall to the ground, right? So essentially, when we're thinking about in a liquid environment, which a lot of my experiments are happening in a liquid environment, if you have something that's essentially heavier than that liquid in the liquid environment, that's going to settle, and that's sort of sedimentation, right? Whereas in microgravity, you don't have these apples falling. If you throw up an apple, it's just going to keep traveling, essentially. It's not going to fall back down. So we have some NASA astronauts here enjoying some fresh food, came up on a resupply mission. These apples are going to stay essentially in suspension, right? So really important, again, for these experiments. On Earth, you have these sedimentation forces with gravity. In space, you have this really nice, even suspension of particles in a liquid. So think about some experiments that might be able to happen. These particles are going to stay nice and evenly suspended. Secondly, we have a lack of buoyancy. So if you have a nice, tasty beverage from the UC Staff Club, the bubbles in that nice, tasty beverage are going to rise to the top. So essentially, we have these buoyant particles, so these buoyant air bubbles are going to rise to the top of this liquid. Whereas in microgravity on the International Space Station, we have air bubbles, or anything sort of lighter than that liquid, are going to mix evenly and stay, again, in that suspension. So on the right here, has anybody seen Don Pettit's Twitter feed, by chance? So Don Pettit is a NASA astronaut. He's spent some time both in Christchurch as well as down in Antarctica. When he was traveling through Tauhere, he happened to pick up just a torus ruler that many of you might have used in school. And so this torus ruler, we're pretty sure, is the first New Zealand-made item that's gone to the International Space Station. So it beat our missions up there. But here, Don Pettit's doing some really cool acoustics experiments looking at sound waves in water and air bubbles. But again, these air bubbles are going to be nice and mixed evenly within that microgravity environment. And then the third important thing for the physics of microgravity is the lack of thermal convection. So if we have a flame here on Earth, we're going to have that heat rising, that lighter air rising and cooler air falling. So this is essentially thermal convection. So these convection currents with heat and cooling rising and falling. Whereas in microgravity, you don't have this thermal convection. So essentially that heat is going to stay essentially right around that flame. So these things are really important. So the even suspension, the lack of buoyancy, and the lack of thermal convection are really important aspects of the microgravity environment for being able to do scientific research. So it's a really unique environment where we can do some really amazing research. But I've probably not convinced you yet, right? So cool physics, so what, right? So hopefully, like I said, by the end of this talk, I'll have convinced you that microgravity is quite valuable. So from my perspective, research on the International Space Station and in the microgravity environment is the ultimate and blue sky research, right? So this fundamental research, this is a whole new environment that we haven't really been able to explore other than sort of the last couple decades. And one of the International Space Station slogans is off the Earth for the Earth. So we're doing research off the Earth, or 400 kilometers up, for the Earth. So the research that we're doing up in microgravity can actually benefit those of us here on the Earth. And so over the last sort of 2.5 decades since the International Space Station has been commissioned, there have been thousands of experiments across a range of research, from materials research and sort of basic fundamental physics research, to microbiology, to plant biology, to human physiology. So the astronauts are actually experiments themselves. And then now, as we're starting to evolve this microgravity research, starting to get a more commercial focus to the research, and there's actually been quite a bit of in-space manufacturing across materials and biological experiments as well, sort of, again, over the last 2 decades. So again, the ultimate science job is getting to be an astronaut and getting to do these experiments off the Earth for the Earth. So obviously we're all here on Earth, but in addition to the research that we can do to benefit Earth, we can also think about the research that we're trying to do to help develop sort of long-term and further space exploration. So we're here on Earth. We're on this tiny little dot, as you can see, Earth from Mars, from the surface of Mars. And so the research that we're doing in the microgravity environment will also help us, like I said, to research and understand the hazards of human space exploration. So things like altered gravity, radiation, isolation, and harsh enclosed environments. The research that we're doing, again, in the low Earth environment, the low Earth orbit environment, can also help us to understand how we can help, become a multiplanetary species, essentially. So if we look at this in a daylight, so this is Mars, so this doesn't look really hospitable, right? So the research that we're doing is going to enable us to help, create sort of off-planet, off-Earth habitation. So how can we turn something, desolate, where there's not really any resources, into somewhere that humans can survive? And when I think about this, I think it's the ultimate experiment in sustainability, right? So if you don't have many resources, you've got to utilize those resources absolutely as efficiently as you possibly can. So this research that we're being able to do off the Earth can also help us to understand how to become a more sustainable species. So one of my favorite things that they do on the International Space Station is they turn yesterday's coffee into tomorrow's coffee. Right? You don't want to think too much about how that works. You've got several crewmates, you're drinking your coffee, you don't want to think too much about where that coffee came from. But there's some incredible technology that's been developed to actually purify all of the waste streams, all of the wastewater that comes out of either the sweat or yesterday's coffee into these water purification systems to recycle about 90% of the water on the International Space Station. So obviously it's quite a bit to get the mass of water off the Earth up to the International Space Station. So having that technology and developing that technology will also help us to become more sustainable with our resources here on Earth as well. So that's what the current space station technology allows us to do. As a biochemist, one of the things that I'm interested in doing is turning yesterday's curry into tomorrow's curry, right? That's a whole different ballgame from turning coffee into coffee. So it's a little bit more in-depth. There's not just a technological machine that will do that for you, but biology will help us do this, to take waste carbon sources and turn that into valuable carbon sources. And so my background is mostly plant biology, biochemistry, structural and fungal biology. And so space to me is just this absolute new frontier of the biology that we're able to do off the Earth for the Earth. And so this is just a little snapshot of some of the things that I would love to eventually be doing. So we can use biology to optimize food production, biomaterials production, environmental monitoring and biosensor development, water treatment, waste management, air treatment, and even habitat development. So we can use fungi to actually create houses and structures and things. So there's just a wide array of things that we can do that just absolutely excite me as to the possibilities of biology in space. But we're going to come back a little bit closer to Earth again. So we're going to focus again on the research that we're doing at the moment. And I say we, this is the consortium of organizations that are working up on the International Space Station. And so I'm a plant biologist by training. I grew up on a farm in a tiny little town in the middle of the country in Indiana in the US. So plant biology is very close to my heart. And so there's some incredible plant biology going on the International Space Station. One of the reasons why plant biology research is so important on the International Space Station is obviously for food development. So the only food that the astronauts are really getting, either coming up from resupply missions, most of that is not fresh, with the exception of those apples that the astronauts got. So most of it is like you would eat if you're going tramping. But having the ability of plants to actually make this food, so there's some hatch green chilies that were grown just a little bit ago on the International Space Station. So learning how to grow food in microgravity is a really important aspect of some of the research going on. Also, atmosphere regeneration, obviously taking in the carbon from the CO2 that the astronauts are breathing, putting out oxygen. Plants are really important for psychological well-being. So it's quite a sterile, kind of stinky environment up on the ISS. So plants are really important for just having something alive to see. And then some really amazing, just sort of more fundamental research. So things like plant-microbe interactions are really important for things like hydroponics here on Earth. How do the microbes in the environment actually help plants to grow? And then a range of fundamental biology research as well. So understanding how plants respond and adapt to this microgravity environment. And then from my perspective, again, getting back to sort of that synthetic biology, how could we use things like algae, which essentially make their own food just using sunlight? How can we use them to engineer them to create food or fuels or pharmaceuticals using just these sort of these microorganisms, these sort of single-celled algae in plants? So lots of incredible plant research. Can't leave out the animals. So animals are the sort of literal guinea pigs up in space. So over the last couple decades, there have been a wide range of animals literally fly to the International Space Station. So everything from fruit flies, like you might have on labs here on Earth, to fish, which is just absolutely incredible, thinking about fish swimming in the microgravity environment. And then quite a few mice that have gone up to the International Space Station as well. So there are nearly 600 mice that have made the journey up to the ISS, so essentially just about as many mice as astronauts. And these experiments are really important for understanding changes in sort of muscular skeletal systems. So without gravity, obviously you're not using your bones and your muscles, so understanding how bones and muscles change in space is really important. cardiovascular, immune systems, circadian rhythms, metabolism, aging, and microbiomes also change in space. So rather than cutting open astronauts when they get home, these poor little mice get to literally be our research subjects. But really important research that's going on. And these are some photos of the mice in their cages on the International Space Station. In addition to plants and animals, some really great infectious disease research that can be correlated right back here on Earth. So some really neat things about microorganisms. So latent viruses become reactivated in space. So astronauts will go up into space not having any viral problems. And then these viruses, so things like viruses that cause cold sores, will actually become reactivated in space. And so you actually, these astronauts have an increased viral load during spaceflight as compared to pre and post-flight. So these viruses become reactivated in space. And so understanding how viruses become reactivated in space can be directly relevant for how viruses become reactivated here on Earth as well. In addition to viruses getting reactivated, we also have increased bacterial virulence in microgravity, and the International Space Station is full of bacteria. You'd be surprised at, basically where they swab, how many bacteria are around. So understanding that increase in bacterial virulence is also really important for astronaut health, but then also understanding bacterial virulence back here on Earth. And really important during COVID, some of the spaceflight developed technologies for saliva collection and rapid viral detection. Obviously, we're quite familiar with those, are now used on Earth as well. So this is one of our NASA astronauts essentially testing out some new viral detection systems. This is one that I'm quite excited about, and this is fairly new facilities that are being developed on the International Space Station. So cell culture in space, obviously without that sedimentation with gravity pressing down on cell cultures, you can actually create three-dimensional cell cultures similar to organs and tissues that we have in our bodies right now. So if you're trying to do this on Earth and you're trying to create these cell culture systems, they'd essentially just all sort of fall flat. And so you can't learn a lot about them. But in microgravity, you can actually study these three-dimensional cell culture systems in a way that we can't do on Earth. And then there's this really cool company called Redwire Space that's developed a biofabrication facility. And they've just recently printed the first knee meniscus in space. So again, if you're trying to do that here on Earth, you want to be able to get the sort of morphology that actually looks like a knee meniscus. But in microgravity, you can print these with a really good sort of robust nature that actually looks like a knee meniscus that you might actually have in your knees. So biofabrication in microgravity is really starting to take off. And then another really exciting one that's really exciting for me is because I got to see this experiment when it first came off of the International Space Station, because the scientists that were doing this research needed to use the microscope that was in our lab at Kennedy Space Center. So I got to see this first. So these are some heart cells that were bioprinted in space. And so they were basically sent up to space, actually printed into a heart tissue, and they came back to space, and we got to see them when they first came back. So they were alive, and they're actually doing their thing. So quite incredible what we can do with cell culture and biofabrication. Now, getting into some of the research that is very near and dear to my heart is pharmaceutical formulation. So I'm now in the chemical formulation design program in the School of Product Design. So I lecture quite a bit about pharmaceuticals. And there's some really neat things that we can do with pharmaceutical formulation. So up here on the left, that little Y-shaped protein is an antibody called Keytruda. And so Keytruda is a really important pharmaceutical for the treatment of a range of different cancers. But at the moment, basically, if you want to get this treatment, you have to go into a hospital, go into a clinic, and get the treatment via IV. But if you have this Keytruda formulation in a crystalline formulation, you can actually administer it via syringe outside of a clinical setting. So it would really open up a lot of opportunities for people that aren't able to get to a hospital or a clinic with regular frequency. But if you try to crystallize this here on Earth, you get some really messy, sort of not uniform crystalline formulations. But if you crystallize this in microgravity, you get a really nice, robust, uniform crystalline formulation. Like I said, we can't actually get this here on Earth. And so if we have these crystallization facilities for antibodies and biological pharmaceuticals, we can actually get new and improved formulations for a range of pharmaceuticals and really benefiting patients, again, to be able to have these formulations outside of the clinical setting. So really cool. And then, like I said, near and dear to my heart. It might not sound as exciting as mice flying through the International Space Station, but protein crystallization. So protein structure is really critical for understanding life. So we are essentially bags of proteins. So our hair, our fingernails, the enzymes that metabolize the sugars when we eat things, basically all of these are proteins. So they're really important in every part of life, and basically every human, or every living thing, from microbes to viruses to humans. And what's really important about this aspect is structure informs function. So if we know the shape of a protein, we can understand how it works. So in the case of COVID, if you guys remember the spiky bits on the outside of the COVID virus, understanding what the structure of those COVID virus proteins are allows us to create vaccines and antibodies and antivirals against the virus. So understanding the structure of these proteins is really important for, like I said, developing these vaccines and therapies. Now the crux of this protein crystallization so protein crystals are used to determine structures. And essentially, we take purified protein. So say we take purified this COVID spike virus, and we use it to grow crystals. And we won't get into exactly how that happens. But essentially, you get these nice, beautiful protein crystals. They look sort of like sugar or salt crystals, really delicate, beautiful things. And then we shine them with X-rays and sort of blow them up a little bit. And then we do a lot of math, and then we're able to get this atomic level structure of these proteins. But the crux of all of this structural biology is the actual protein crystal. And these can be very difficult to actually grow in gravity. So it often requires screening hundreds to thousands of different crystallization conditions. And for the scientists that do it, it's as much an art as a science. So you need to have this high throughput crystal screening to actually get a crystal that might work to get a nice structure. So lots of labor, lots of money, and for often inconsistent results. This is where microgravity comes in, and I could have used this when I was going through my career a decade or so ago. Is protein crystallization in microgravity. We've been doing this since the shuttle eras, and you can actually essentially get much larger microgravity crystals and much more uniform protein crystals as well. So sort of what we saw with the antibodies in Keytruda. So you get much larger, more uniform crystals, essentially. So you can see here, again, some more antibodies that were grown by the pharmaceutical company Merck, much bigger crystals in microgravity than you can get on the ground. And so these are a couple astronauts doing these crystallization experiments. So again, it still takes a lot of time. Astronauts are not cheap, but some really, really great research that can be done. Really cool sort of a meta-analysis of all of the crystallization experiments that have been done that are publicly available. You get much larger crystals, you get much structurally better crystals, so they actually survive the X-rays. You get more uniform crystals, and you get what's called an increased resolution limit, so you can actually get a better picture of exactly what these structures look like, and then improved mosaicity, which we're just going to say essentially it's another quality aspect of these protein crystals. And so about 90% of crystals that we've tried to grow in microgravity are better than ones that we can grow on Earth. So again, really valuable microgravity environment for getting structurally bigger, much better protein crystals that can help us develop vaccines and therapies. Right? So keep this in mind, really valuable protein crystallization in microgravity that we can't do on Earth, but takes a lot of screening in order to do that. All right, so microgravity research. Hopefully we've seen that there's a lot of great research going on. Again, there have been thousands of biology experiments over the last 20 years that have led to clinical applications, life support systems, pharmaceutical development, and even in-space manufacturing of these new formulations. But it's not perfect. So again, there's limited space, limited hardware. Obviously, you can't just build a whole research lab up in space. Limited experimental options, and again, both limited and expensive crew time. So again, these astronauts, basically their schedules are basically in 5-minute increments, and about 200,000 US dollars per hour for astronaut time. So if you want to get an experiment done, it's going to be quite expensive to get it up and get the astronaut to actually do it for you. And then sort of the elephant in the room is that the ISS is due to be decommissioned in 2030. So we've got all this incredible research. How are we actually going to be able to do this after 2030? And this is where commercial space comes in. So it's a really exciting time to be a part of sort of commercial microgravity research, because there are 4 different either companies or consortiums of organizations that are actually building commercial space stations. So we have Starlab, Haven 1, Orbital Reef, and then I am absolutely not biased whatsoever. The best one is Axiom Station. So we're actually partnered with a company in Houston called Axiom Space, who will be building the first commercial space station that will actually be essentially built onto the ISS, and once they have their 5th module, their power module, they'll decouple from the ISS and become a free-flying commercial space station where we can do basically all sorts of incredible biological and other research in microgravity. So like I said, New Zealand has a partnership with Axiom Space, but there are several other companies that are also building commercial space stations, which really open up the opportunities to be able to do more of this research, and not only for a fundamental reason, but also for commercial research as well. So this is my friend Lucy Lowe, who's the chief scientist of Axiom Space. We actually met 20 years ago in a science camp, and she just happens to be the chief scientist of Axiom. And so we're just checking out the sort of bedroom module of Axiom Station right there. So tiny little bedroom if you want to become an Axiom astronaut. All right, so where do we fit into this? So how many of you know that New Zealand has a space agency? Fantastic. So the New Zealand Space Agency was established back in 2016, essentially as a result of Sir Peter Beck wanting to launch rockets from New Zealand. So back in 2017, Rocket Lab launched their first orbital class rocket, so the Electron rocket. And so that was essentially sort of the dawn of the space industry here in Aotearoa. If you're wondering how a biochemist gets into space, like you all, I've always been interested in space. I threw my name in the hat for NASA's astronaut selection back in 2017. Was really fortunate to make it from 18,000 applicants down to the finalist interviewee stage, and really just opened up my eyes to the value of being able to do this research in microgravity. Didn't quite make it to the top 12, but basically the day after that first Electron launched was the day after I found out that I was not going to be an astronaut. So I decided, what can I do to help develop the industry here in New Zealand? So we're really fortunate to get some research funding in 2019 to start looking at protein crystallization on CubeSats, so in these tiny little satellites. We had some couple issues with trying to get a good satellite partner. And then in 2022, we established our partnership with Axiom Space to get things to space stations. We finally did launch our CubeSat, but unfortunately, we didn't establish contact with that. So unfortunately, the CubeSat experiment sort of put the kibosh on that. But then we got some additional research funding late last year. So a 5-year program, $10 million, to essentially start building out commercial microgravity research. Again, focused mostly on this protein crystallization. So we had our first mission to the space station, so New Zealand's first mission to the space station just earlier this year, which I'm going to be talking about, and then we've got a couple other launch missions planned in the next couple years. and then hoping to have permanent commercial facilities on Axiom Station by 2028. But you got to start small. So we've literally started small. So we had New Zealand's first mission to the space station. So I didn't bring Lucy here. So Lucy is our protein crystallization prototype, which we'll talk a little bit more about. And then we had a second bonus payload that was not initially supposed to be in our manifest, but through a range of happy accidents, we got some more payload space. So I'll let you guys come up and touch something that's been to space if you'd like to after the talk. So the mission was coordinated via Axiom Space. We launched on the 21st of March for a 38-day mission. This is on SpaceX's commercial resupply mission 30 out of Kennedy Space Center. So just a little bit about Lucy. So Lucy is essentially taking a biochemistry lab and shrinking it down. So essentially we have a microscopy facility and about 200 different wells of protein crystallization experiments inside Lucy with autonomous image collection and autonomous environmental monitoring. So we can essentially see real-time protein crystallization as it's happening. So Lucy, what we wanted to do, make sure that she survives launch in microgravity, that we can communicate with her in orbit, that we get that daily image generation, and we have that autonomous monitoring of the conditions, and that she survives deorbit and the return back to Earth. Our second, like I said, our bonus payload, we ended up essentially stuffing about 30 different protein experiments into this payload, so mostly looking at protein crystallization, and also we had some radiation experiments as well. We had proteins from 4 different universities from around New Zealand, as well as my partners at Arizona State University. So these proteins are involved in infectious disease, in cancer, and in sort of some really cool biosynthesis of some high-value compounds. We also had some microfluidics experiments to understand what the best housing for our protein crystallization experiments is going to be. Main success criteria for our crystal screedy payload, that again, they survive launch and return. We get some initial data, and we get an initial understanding of what this experimentation in microgravity is all about. So for all of these experiments, we had basically one box in space with about 30 experiments, and another box that was sitting in an incubator back in the lab, so we can compare the ground controls to our microgravity experiments. Getting to go to the Kennedy Space Center was an absolute dream come true. How many of you have ever been either to Houston or to Kennedy to get to, yeah. Man, I get goosebumps thinking about it. So getting to go, we had our own lab there at the space station processing facility, and with our team here from New Zealand and Arizona, getting to be there with the team, to assemble the payloads, to assemble all of the science, and then hand it over to our cold stowage team was just an absolute dream come true. Really late nights, really early mornings, lots of mahi, but being able to be there with the team and actually see these experiments get essentially loaded into a cargo Dragon on top of a Falcon 9 rocket was, just absolute dream come true. In addition to the mahi we're actually doing, Kennedy Space Center is just an incredible, both a spaceport as well as a wildlife and a conservation area. So the wildlife, we got to see manatees, so many alligators. I didn't get to see any snakes, fortunately. Herons, the bird life, just there on the Cape, on Cape Canaveral, just incredible. Like I said, both a spaceport with dozens of launch pads, but then also this wildlife. And what it really did was remind me of what we're doing out at Kaitariti Spit. So Tauhere joint venture is a joint venture between the Runanga out at Kaitariti Spit and the New Zealand government, with dual kaupapa of developing a spaceport and wildlife conservation. And it really reminded me of what NASA has done out at Cape Canaveral. And so really excited about what we can do here in Aotearoa. Getting to see the rocket launch, like I said, I still get goosebumps. So this is our rocket. So this is commercial resupply mission 30. We were not very far away. So we're at the press viewing area, just outside of the vehicle assembly building. I didn't cry. I thought I was going to cry. We have our 2 payloads, New Zealand's first payloads, going up to the International Space Station. Again, being able to watch with our team that had put years of blood, sweat, and tears into the experiments and actually see it lift off the pad was just, like I said, an absolute dream come true. So New Zealand's first mission headed up to the ISS. Basically I stayed nervous until a couple days later when the Cargo Dragon actually docked to the International Space Station. So when it actually latched on and Mission Control said we're all good, I started to breathe a little bit and we were able to come back to New Zealand for the commissioning of Lucy. So Lucy was plugged into power and data, and so we were able to start getting our initial crystallization data and images down essentially once everything was plugged in. So this is up on the ceiling essentially of node 2, which is just outside of the crew quarters. And so Lucy got to spend 38 days. The protein crystal screening plate was in an incubator, so we didn't get photos of her up on station. But stayed up there for 38 days, came back down, and again, myself and collaborator Alexander Ross from Arizona State University were like two kids on Christmas, waiting for the splashdown in the Gulf of Mexico, and then to actually get to analyze the science that came back down. I'm not going to talk too much about the data itself. You'll have to wait for the publications for that. But we were able to take our protein crystal data and put these in X-ray synchrotron and actually start to get some structural data. So really exciting results. You'll have to wait to the next talk for that. Successes that we have from the first mission. So essentially we understood how to develop a mission. So again, this is first mission from New Zealand. So going through that mission development process, the launch and return process for the first time, so we understand how to do that. So if others here in Aotearoa are looking to do that, very happy to help set these missions up. From Lucy, we got some really great data for our hardware and control systems, worked flawlessly in orbit. So we got, again, this real-time data, which gave us really critical flight heritage of our subsystems that really are informing our next module development. Our crystal screening plate, basically everything came back intact. Again, opening up this little box was what we called anxious Christmas, but everything was intact. We took about 30,000 images when that came back, so it really hit home having sort of automated systems, really brought home the value of what we're doing for developing a high throughput crystal screening service that is fully automated, because doing this when it came back was days and days and 30,000 images trying to get everything processed when it came back. But yes, we got some really exciting experimental results comparing our microgravity experiments to our ground control experiments. So again, that will come out soon. Stay tuned. So a little bit of suspense for exactly what we found. But yeah, like I said, understood the value of what we're developing, understood the need for a comprehensive ground control testing and optimization, which we're going through at the moment for our next modules. And then one that we weren't really necessarily expecting, but it's things that I'm telling the space agency. So obviously, crew time is expensive, but also really valuable. So we weren't able to get a lot of images and video with our experiments on station, which didn't affect the science, but it would have been quite kind of nice. But if we had some Kiwi astronauts, they might be invested in getting some photos and videos. So if you see anybody from the New Zealand Space Agency, tell them Kiwi astronauts invested in the payloads that are going up would be really beneficial to missions. And if you guys want to go, I will vouch for you, and then everybody wins. No, but in all seriousness, we're really excited about the opportunities that our first mission has enabled. And so we're now, like I said, off and away developing our commercial microgravity protein crystallization platforms, essentially for the pharmaceutical and biotechnology industries. So we're right in the thick of hardware, software, and subsystem development for our next missions towards full module design, fabrication, and testing. So we'll have flight heritage via 3 missions. So we are our prototype Lucy, just flew this year. We'll have our minimal viable product flying in 2026. We hope to have some commercial clients on board. And then we hope to have our commercial unit flying sort of all the bells and whistles, fully automated, high throughput protein crystal screening service on Axiom Station or the ISS from 2028. And then also looking beyond protein crystallization, so this is a really valuable thing that we can do for revenue generation and for data generation. But obviously there's a lot of things that we can do here in Aotearoa. And so if anyone is interested in expanding missions beyond protein crystallization, we're starting to expand. And so if anyone is interested, please come talk to me, looking to develop a network of researchers, or just people that are interested in things that we can do in microgravity. So New Zealand does have quite a competitive advantage. So we do have unprecedented access to orbit. I thought we were the 4th, but I think we've now become the 3rd most frequent launch country in the world. We have 2 launch providers. We obviously have Rocket Lab from Mahia Peninsula, and Dawn Aerospace is developing orbital capability with their space plane as well. In addition, we have partnership with Axiom Space, which gives us access to the International Space Station and then a commercial space station once they're up and running. Really, especially coming from the States, one of the real values that I definitely have experienced here in New Zealand is we have a very collaborative domestic aerospace industry. As a biochemist, I would not be able to do what I'm doing without a collaborative team and a really great network of engineers and scientists around New Zealand that are willing to work together. And that includes the New Zealand Space Agency. So we have a really streamlined regulatory process, both for launches, but as well as for payload permits to actually get things up onto space stations. And it's really nice when you know the people and you can just call them up. It's a little bit different than working with NASA, who are great, by the way. But I don't know, I basically know everyone in the Space Agency. I obviously don't know everyone in NASA. And again, some really great connections with both the domestic and international biotech and pharma industry, who will all be our clients. And then one that I really cherish and value is our vision Mātauranga and Mātauranga Māori, our unique values-based policies, knowledge systems, and just the communities. So working with Tawhaki Joint Venture out at Kaitoriti Spit has been a huge, just something that I'm really grateful for. And so seeing us develop a spaceport less than 50 minutes from here, if we can get Dawn Aerospace to bring my experiments to space station and bring them back down, would be absolutely phenomenal. But again, just real competitive advantage. And I would say another competitive advantage is just our incredible team. So these are some of the people that have been essentially helping drive the work for both the first mission and as we go forward. So from here in Otatahi, we've got David Wright of Asteria Consulting, Dave Sanders of Intranel, Volker Knock from here at Canterbury, Sarah Wickie-Bennett was one of our Kaiahi Māori here at UC, and then Cather Simpson from Auckland, Alexander Ross from ASU, and lots of great people at Axiom, including Lucy and Jana. And just quickly, these are a few of the researchers that had research on our mission from Waikato, Otago, Wellington, Auckland, and then UC. And again, just an incredible international team that I have had the joy of working with. And finally, for all of you here in the audience, I just want to thank you for your incredible attention. And this has been my dream, and this is the dream that I get to help continue with this great team. But for all of you, the future belongs to those who believe in the beauty of their dreams. And so if your dreams do involve microgravity or anything up in space, love to have a chat and very happy to take any questions. Thank you. Brilliant. Go for it, mate. I will have mission patches down here.
Ah, great. Yes. First of all, can I have a mission patch?
Stickers, yeah.
My name's Josh. I'm a planetary geologist, so my vote is Mars. But and we did see a bit of your sort of future plans there, but thinking even further and even bigger, if I gave you $10 billion, and it was real money, US dollars, what would be the top of your priority list if I told you you had to spend it on space?
Life support systems. So from my perspective, again, where my real passions lie, or how do we engineer organisms to produce pharmaceuticals and food and those sort of resource generating biological systems. Yeah, so let me know. We'll sign the check. Yeah? Yeah.
I'm asking, who was the first, who came on the Earth? Who was the first one?
Well, the question was who came on the Earth or who came off the Earth?
On the Earth.
Off the Earth? Yep, that's a good question.
She asked before who came up with these ideas, which obviously a very open question. So
Yeah, who came up with these ideas? That's really, really good question. So all of us came up with these questions. So people that are really curious, people who have questions. If you have questions and you're curious, you can become a scientist and you get to do some of these experiments. So to answer your question, anyone that's curious and has questions and want to find out those answers to questions, you can become a scientist and do some of these experiments yourself. Thank you.
What made you want to study science in the first place, and did you ever think you'd end up doing something like this?
Excellent question. So the question was, what initially got me into science, and did I ever think I'd be doing, research in space? So I don't know how many of you guys have had a teacher in your lives that really changed your life, and that teacher for me was Miss Sharon McElroy. So I was an athlete. I didn't want to become a nerd. I wasn't interested in becoming a nerd, even though I was a nerd. And I didn't really want to get into science, but Miss McElroy was running a science research program at my tiny little school in the middle of nowhere in Indiana. And she said that my friend won $25 in a science fair. I was like, cool, sign me up. But no, I've always been curious as well. And so growing up on a farm, I always wondered, how do plants grow and how do plants interact with the environment around them? So I created a science research project, essentially looking at sort of how different plants and trees were growing on our farm. And that basically led me into a plant biology degree as an undergraduate. And then I really enjoyed that, so went on to a PhD. And then that brought me here to New Zealand. And again, as well as science, I was always interested in team sports and adventure. And the combination of that as an astronaut really, really excited me. So that's what led me to apply to be a NASA astronaut. And that's sort of what led me to where I'm at. So a lot of careers, aren't necessarily straight lines. There's a lot of weaving in and out of different things. But really, I guess the thing that ties it all together is being, like I said, being curious and wanting to find out those answers. So I didn't think that this is where I would be, but I absolutely love what I'm doing right now.
Hi. What were the major design considerations and what sort of tests did you have to conduct on Lucy to make sure it would be reliable throughout the whole mission?
Absolutely, really great question. So the question was, what were some of the design considerations that went into essentially making Lucy? So Lucy was originally designed for a CubeSat, so actually in the vacuum of space. Essentially a CubeSat's a tiny little, essentially a free-flying tiny little satellite that is exposed to radiation, is exposed to the vacuum of space. And so a lot of the design constraints were for CubeSats themselves. So designing to be radiation hardened, so essentially so that radiation can't kill your computer, and that the heat and the cold, depending on where you're at in the orbit, so understanding environmental both monitoring and then control, and then obviously sealing for the vacuum of space. So when we basically shifted from CubeSats to the space station, all of our design considerations were absolutely fine for the comfort and essentially environmentally controlled environment of the ISS. But essentially mass and volume are real big constraints as well, so trying to get things as small and robust as possible. And if you want to get to more of the technical things, happy to have a more in-depth chat down here. Great question.
Here and then
Absolutely. Yeah.
So as you've shown, the microgravity environment's essential for growing some of these protein crystals, but something I was wondering was, does the motion of the spacecraft affect how those crystals grow? And also, is there any challenge bringing them back from space on the high G forces and whatnot?
Excellent question. So the question was essentially does the motion of the space station affect the growth of the protein crystals, and what about essentially the high G forces and vibrations and things of getting these fragile protein crystals from orbit back down to Earth. So the motion of the ISS is not really a problem for the protein crystals themselves. So again, you're within the pressurized environment of the ISS. The thing we do have to worry about is vibration. So if you've got the experiments essentially sitting next to sort of the stationary bike, and you've got lots of vibration, that vibration is going to affect the protein crystal growth. And then obviously the G-forces and the vibration, once you've got these beautiful fragile crystals coming back from space station, is definitely something to take into account, which is why we're designing the microfluidics essentially to help mitigate some of that. But yeah, definitely something to take into account that we are mitigating with the environments that we're developing.
Hi, I'm Team Mars, and I think I've been contemplating, because I'm getting a bit older and I would like to do a one-way trip to Mars eventually in my 80s. I was thinking when and if I die up there, instead of burying me, they should burn me and put carbon into the atmosphere to help start the terraforming of the planet. Could that be a possibility for dead bodies up there to help with terraforming?
There's a lot to that. Yeah, so thinking about, I mean, like I said, there's a lot to that. So thinking about one-way trips to Mars and what happens to humans. So that is both a sort of in situ resource utilization question, and then also there's a lot of values questions about, what as a society, as a world, as a human race, what should we do? And that's definitely something, a philosophical question to think about that is beyond the scope of, I guess, biological experimentation, but cool to think about. Thank you.
Yeah, similar question. I'm interested about the last sentence you mentioned, the value-based policy. So do you do like a moral test for the scientists to go to the space if they commit a crime? Who's gonna policing the stuff? And also, imagine in the future if every country can have a mirror colony in Mars or somewhere. Maybe New Zealand will have a New Zealand point too.
Yeah.
Yeah. Do we still need like Treaty of Waitangi?
That is an excellent question. So the question was around sort of values of low Earth economy and sort of what is the jurisdiction of Aotearoa beyond sort of our economic zone here on planet Earth. Is that so? Yeah. Again, very good philosophical questions that people are more sort of politically minded are probably working on than me with just the scientific research. But again, definitely something to think about. So there are international treaties that govern space. Similar to that, there's international treaties that govern Antarctica and sort of shared resources. And so it is really important that we bring our values from New Zealand as a country to that international and global sort of both the organizations and the agreements as well. Yeah. Again, so not just for scientists, space is for politically minded people as well to help develop these international agreements. Thank you.
Hi, thank you. That was a fascinating talk. Question about how space now is trending towards kind of a commercialization and privatization, coming from NASA, ISS now to private commercialized space stations. How do you think this will affect science in the next 20 years or so? And how can that change what you're doing? Will it affect it positively, negatively, you think?
Yeah, great question. So the question was, as we shift from more governmental research and governmental space stations and organizations towards commercial space stations and organizations, how does that change? So there definitely will be a lot of changes. So at least with Axiom Space, obviously, it's a commercial company that's looking to make revenue, both from astronauts and then the science that's going to be done. And so it will shift what we're doing from a lot of fundamental research towards that commercial revenue generation research, but there will be opportunities for subsidized, more fundamental research as well. So the focus will be, how can we generate revenue from these research partnerships and organizations. But I don't think that will fundamentally change the value of what's coming out of that. And there will still be governments that essentially utilize those commercial space stations. So NASA is counting on the commercial space stations to be able to send fundamental research up as well. But it will be interesting. It's literally a whole new frontier. So we'll see sort of how everything shakes out over the next couple decades.
Just letting you know, we've got plenty more questions, but we probably only have time for 2 more.
Okay.
But feel free to.
Yeah, I'll be here.
Ask your questions at the end.
I was just wondering when the results from your research come out.
As soon as I can get to. So the question was, when will the public results of the research come out? Essentially, as soon as I can get time to analyze all of them. As an academic, there's a lot of responsibility, so I'm really looking forward to a summer where I can actually dig down into these 30,000 images that we've generated. So stay tuned. I'm really excited to get that out. So yeah, hopefully in the next couple months. Yeah. Cheers.
Last question.
Last question?
Solve that one.
Great speech. Thank you very much. You talked a lot about costs and the amount of money that's been spent, and you're trying to do all this stuff in a microgravity environment. Can we not create one here on Earth?
Can we make microgravity here on Earth? Is that the question? Yes. So in theory, yes. So if you were to drop Lucy, our beautiful payload, from the ceiling here and drop her down to the Earth, that couple of seconds, she will experience microgravity. But for the protein crystallization experiments, you really need that long duration microgravity. And so there are sort of analogs of microgravity. So you can have essentially random positioning machines that essentially rotate things. You can have, there are things that sort of simulate microgravity, but not to the extent of that not sedimentation, no convection forces, things like that. So for the experiments that we're trying to do, you can't simulate that environment on Earth to get these protein crystals, to get these crystallization conditions that you can in microgravity. Other experiments you can, but again, they're very small scale and not exactly analogous to what you'd experience 400 kilometers up. No. Fantastic. Thank you all so much I will be sticking around, but otherwise, thank you and have a great rest of your evening.